Hello everyone, welcome to this lecture on cell membrane transport and today we are going to understand simple and facilitated diffusion. So then we are going to start with understanding the various chemical compositions in the extracellular and intracellular fluids. Then we will see the structure of cell membrane and some important proteins that are present in the cell membranes. And then we will understand the difference between diffusion and active transport, and then we will see the simple diffusion and facilitated diffusion, what these are, and some important examples of channel proteins, voltage gated and ligand gated ion channels. Then we will understand in detail the facilitated diffusion, and we will end with understanding the various factors that affect the net rate of diffusion. So then let's start with the chemical composition. So as we can see here that the extracellular fluid contains a large amount of sodium, okay, but only a small amount of potassium, okay. So these two ions are very important to know about, as well as you can see that there is large amount of chloride ions too here, as well as phosphate ions is on the higher side in intracellular fluid, while bicarbonate ion is on the higher side in extracellular fluid. Now let's see the cell membrane. Now as we know that the cell membrane is a lipid bilayer, okay, and there are large number of protein molecules also, as you can see here, and these protein molecules are very important for the cell membrane transport, and two such important proteins are the channel proteins and carrier proteins, okay. So what are these channel proteins? These channel proteins, as you can see that there is a path here, okay. So this path contains water, so these are watery spaces here, okay, just like river channels, okay. So these channel proteins contains river channels like watery spaces all the way through, okay, and this watery space allows free movement of some ions or molecules, okay. While these carrier proteins, as you can see, what happens here is that when some molecule or ion bind to these carrier proteins, it leads to some conformational changes in these proteins, okay. So when some molecule, if it wants to enter inside the cell, it will bind to this carrier protein, and when it will bind, it will lead to some conformational change, and this conformational change will help in the movement of that molecule or ion inside, okay. And as you can see that these carrier proteins are of two types. First one is involved in the facilitated diffusion, while these two are involved with the help of energy, okay. So these two type of proteins requires energy for the movement of the molecules or ions, while this one doesn't require the energy, okay. So the movement of molecules or ions, if it is by the means of simply diffusion through the cell membrane, it is known as simple diffusion, as well as through the channel proteins, it is also known as simple diffusion, while with the help of carrier protein, it is known as facilitated diffusion, while with the help of energy, it is known as active transport, okay. And the simple diffusion and facilitated diffusion combinedly are two types of diffusion. So then passive transport or the diffusion is from the high to low concentration gradient, okay. So the passive diffusion or simple diffusion is like this movement from higher to the lower concentration, as well as facilitated diffusion is also from the higher concentration to lower concentration, but with the help of carrier proteins. Okay. So then diffusion, if we will see the difference between diffusion versus active transport, then diffusion is random molecular movement. Okay. So in there, the molecule moves randomly, and the kinetic energy which that molecule have, that kinetic energy helps in the movement of that molecule, and this is known as diffusion. Okay, while active transport, we saw that it requires energy, and the movement in the active transport is from low concentration to a higher concentration, and that is why, because the movement is from lower to higher concentration, active transport requires energy, as well as there is involvement of a very important carrier proteins also. Okay, so these requires energy besides the kinetic energy of the molecule or ions. Now for the movement of the molecule through the lipid bilayer, it should be highly lipid soluble. For example, steroid hormone or carbon dioxide or oxygen, these are highly lipid soluble, so they easily simply diffuse through the lipid membrane. As well as there are some important protein pores, and these protein pores are known as aquaporins. Aqua means water, so these aquaporins are for the movement of the water. Okay, so they selectively permit rapid passage of water, and the pore is too narrow. It is too narrow to permit the passage of any hydrated ions. Okay, so that is about aquaporin. Now we saw the simple diffusion through the cell membrane, as well as we saw the aquaporins. Now we will talk about the channel proteins. Okay, so there are some channel proteins which are known as voltage gated ion channels. Okay, so they are for the movement of ions, and they are voltage gated, means when there are some changes in the voltage, the gate opens and there is movement of ions. Okay, so they are sensitive to the changes in the electrochemical potential across the membrane. Okay, so three very important examples are voltage gated sodium channels, voltage gated calcium channels, and the most abundant one, voltage gated potassium channels. Okay, so they are most abundant in our body. For example, let's see, these are voltage gated sodium channels. So when there is membrane depolarization, these gates will open. Okay, and when these gates will open, sodium will move, of course, from outside to inside, because sodium ion concentration is higher in this side. Okay, as well as you can see the potassium channels here. These potassium channels also opens when there are changes in the electrochemical potential, and when the gate opens, the potassium moves outside because its concentration is higher in the intracellular fluid. Okay, so the sodium channel and potassium channels, these are very important voltage gated ion channels which are responsible for the propagation of action potentials in nerve and muscle cells. Okay, now we will see one more channel protein that is also very important, just like voltage gated ion channels, and this is ligand gated ion channels. Okay, so here the ligand, when binds to the ion channels, these channels will open. One such ligand is a very important transmitter in our body, it is known as acetylcholine, okay, or ACH. So this ACH or acetylcholine binds to the ACH binding site of the acetylcholine receptor, okay. So when 2-ACH binds to this acetylcholine receptor, this receptor will open, and when this receptor opens, sodium will move from inside. Now this receptor, this acetylcholine receptors is very important for the transmission of nerve signals from one nerve to another nerve and from one nerve to muscle cells, okay. Now we will see the facilitated diffusion. Okay, we saw the simple diffusion, we saw the passes through the membrane, simply passes through the membrane, as well as we saw the channel proteins, the voltage gated ion channels, the ligand gated ion channels. Now we will see the facilitated diffusion. As you can see that the simple diffusion, in this graph the rate of diffusion increases in the simple diffusion, okay, when the concentration of the substance increases, okay, while in the facilitated diffusion, which requires what, which requires carrier proteins, so when the concentration of the substance increases, the rate of diffusion also increases, but it comes a time when it reaches the maximum limit, okay. So here the rate of diffusion is maximum, after that the graph is straight, okay, plateau phase is there. So this part, this maximum concentration, okay, in the facilitated diffusion, the rate of diffusion when it reaches the maximum, it is known as Vmax, okay. This is known as Vmax when the rate of diffusion is maximum, okay. Why this happens, we will see this. So now you, as you can see that in facilitated diffusion there is involvement of carrier protein. So when a molecule which wants to enter inside, it will bind to the binding site, and when it binds to the binding site, it leads to what? It leads to the conformational change, and when this conformational change happens, this molecule will enter inside, okay. But after that it will again returns to its basic structure, okay. So this happens at a rate, and the rate at which molecule can be transported by this mechanism can never be greater than the rate at which the carrier protein molecules can undergo change back and forth between its two states, okay. So this state and this state, it happens at a particular rate, and when that rate reaches at its maximum, it is known as Vmax. After that the graph is in plateau state. Now we know that the facilitated diffusion is from the higher concentration to the lower concentration with the help of carrier protein. Now one important example of this carrier proteins is the SLC transporter or the solute carrier transporter, okay. So the solute carrier transporter is a family, it is known as solute carrier transporter superfamily, okay. And one very important example of this solute carrier transporter is the glucose transporter or GLUT, GLUT, okay. So this is a very important transporter that involves in the transport of glucose molecules in various tissues, okay. So just for fun, I am GLUT. Now we will see the factors that affect the net rate of diffusion, and we know that one important factor is the concentration difference. So CO is the concentration outside and Ci is the concentration inside, and this diffusion is directly proportional. This net diffusion rate is directly proportional to CO minus Ci, okay, that is the concentration difference across the membrane. Now another factor that plays role in the rate of diffusion is the membrane electrical potential. So as you can see that the concentration of these negative ions in the outside and in the inside is similar, but still there is movement of these negative ions towards inside. Why? Because we had applied negative charge outside and positive charge inside. So this positive charge will attract the negative ions towards inside. But after some time, when a large quantity of these negative ions have moved towards inside, okay, and the concentration difference is rises high enough, so these two forces, this concentration difference and this membrane electrical potential difference, these forces, these effects will balance each other. Okay, so after that the movement of ions will goes into the equilibrium phase. Okay, now another factor that plays role is the pressure difference. So where there is a higher pressure, of course there will be the movement of the molecules or ions from the higher pressure side to the lower pressure side. Okay, one very important example is the capillaries. In the capillaries there is 20 mmHg greater pressure inside. Okay, so there is movement from the capillaries to the outside because of this higher pressure in the capillaries of 20 mmHg from the outside. Now we will see osmosis. We all know what osmosis is. This is pure water and this is sodium chloride solution. So there will be a movement of water from this side to this side because of the concentration difference. So the osmosis is the process of net movement of water that is caused by a concentration difference of water. We all know that. But what is this osmotic pressure? If we will apply the pressure on the sodium chloride solution side, then the osmosis of water into the solution would be slowed or stopped or maybe even reversed. Okay, so when we will apply pressure from this side, from NaCl side, then the net movement will be slowed down, okay, or it may be even stopped or even reversed. This is known as osmotic pressure. Okay, so the osmotic pressure is the amount of pressure that is required to stop the osmosis. It's called osmotic pressure. Okay, thank you. Do subscribe, like and share. You have an assignment. You have to share in the comment section that which are the main glucose transporters or GLUT in pancreatic beta cells and in skeletal muscles, and what are their roles. These are the references. Thanks a lot.